what if I told you we can rewrite the very code of life? Take a gene from one animal and just pop it into another. It sounds like something straight out of a movie, right? Well, it's not. Today we are diving deep into the absolutely incredible world of transgenic animals. Okay, take a look at this. I promise you, this is not a special effect. These mice, they are genuinely glowing. And it's all because scientists took a single gene from a jellyfish. Yeah, a jellyfish, and put it into their DNA. It's a pretty stunning picture, and it immediately makes you ask, right? First, how on earth is this even possible? And second, what's the point? Why would you do this?
So the science that makes this happen is called transgenesis, and is actually a pretty straightforward concept at its core. A transgenic animal is just an animal whose genome, its genetic blueprint, has been changed on purpose to carry a gene from a totally different species. The key thing here is that this new gene gets woven right into their DNA, so it can be passed down from one generation to the next.
All right, so here's the plan. We're going to start with the early breakthroughs, the stuff that really did feel like science fiction. Then we'll look at the modern genetic toolkits scientists have now. After that, we'll get into the really important stuff, the life-saving applications. And finally, we'll talk about the big challenges that still lie ahead.
So, how did we even get here? Where did this whole journey start? Well, the story of modern transgenesis really kicks off with one single groundbreaking experiment. An experiment that absolutely captured the world's attention.
For centuries, if you wanted to improve an animal's traits, you had one option: selective breeding. It was slow, painstaking work. But then, the 20th century just changed everything. By 1974, scientists were creating what they called chimeric mice by mixing cells from different embryos. Kind of wild, right? But the real game changer, the moment that put this science on the map, that happened in 1982.
And this is it. This is the image that screamed the news to the world. It was on the cover of the journal Nature, one of the most prestigious science journals out there. What you're seeing is the result of that 1982 experiment. On the left, you have the transgenic supermouse, and right next to it is its normal, everyday sibling. I mean, just look at the difference. The visual impact is absolutely staggering.
So what did they actually do? The method was surprisingly direct. A team of scientists took the gene responsible for growth hormone out of a rat and, using a tiny needle, injected it straight into fertilized mouse embryos. And when those mice were born, boom! The rat gene switched on, and it caused them to grow to almost double the size of their brothers and sisters. It was incredible.
And let me tell you, this wasn't some quiet little lab result that nobody noticed. No way! The impact was huge. It was dramatic. You can't miss it proof that we could fundamentally change an animal and do it predictably using a gene from a completely different species. It lit a fire in the scientific community and, well, sparked a ton of excitement and debate everywhere.
Okay, so that giant mouse experiment basically kicked the door wide open. It was like the proof of concept that showed everyone what was possible. And today, scientists have moved way beyond that. They now have a whole toolkit full of sophisticated methods for creating these transgenic animals.
And that toolkit has really grown. I mean, you've got DNA microinjection, which is a more refined version of what they did with the giant mouse. But now, scientists can also use things like retroviruses, basically using a virus as a tiny delivery truck to carry the gene into a cell. They can even do sperm-mediated transfer, or use something called electroporation. There are a bunch of options, and the one they choose really depends on the animal they're working with and what exactly they're trying to achieve.
Let's zoom in on one of the most common methods, DNA microinjection. You can see it here in this diagram. It's pretty much what it sounds like. You use a microscopic needle to physically inject the gene you want right into a fertilized egg. Now, it's a foundational technique, for sure, but it is incredibly delicate work. The success rate can be pretty low. Still, because it's so direct, it's the go-to method for a lot of projects.
Okay, now for another really powerful technique. Somatic cell nuclear transfer. This one is a bit more complex. Basically, you take a regular body cell, like a skin cell, and you modify its genes in a lab dish. Then you take its nucleus, the part with all the DNA, and you transfer it into an egg cell where the original nucleus has been taken out. That process creates a whole new embryo. And if any of that sounds a little familiar, well, it should. It's the exact same basic idea they used to create the most famous clone in history, Dolly the sheep.
All right, so we've talked a lot about the how, but that brings us to the really big question, right? The why. Why go through all this incredibly complex and difficult work? Well, the answer is pretty amazing.
It lies in the incredible, real-world applications that stretch all the way from our farms to our pharmacies.
Let's start with agriculture, where the benefits are super tangible. Think about it. We could engineer cows to produce milk that's already lactose-free for people who are intolerant. Or what about creating livestock that are just naturally resistant to common infections? That would mean using way fewer antibiotics. We can even tweak sheep to have more muscle and less fat, producing leaner meat. These are not small changes. They're potentially massive improvements to how we produce food.
And if you think that's cool, the medical applications are frankly astounding. By putting specific human genes into animals, we can get them to produce really complex, vital proteins for us, often right in their milk. For instance, pigs and sheep have been engineered to produce human protein C, which is a critical drug for preventing blood clots, and rabbits can make a human protein used to treat osteoporosis. It's just incredible.
This whole process has a pretty clever nickname, farming, with a PH. And that's exactly what it is. We're essentially turning these animals into living factories, or bioreactors. They can produce these incredibly complex medicines for us that are just way too difficult, or sometimes even impossible, to make in a traditional lab.
And the science is definitely not standing still. One of the most mind-blowing frontiers right now is something called xenotransplantation. That's a big word, but the idea is huge. Scientists are genetically modifying pigs to make their organs, like hearts and kidneys, compatible for transplant into humans. Think about what that could mean. It could potentially solve the massive organ donor shortage we have worldwide.
Okay, but let's take a step back. As powerful and exciting as all this technology is, it is not easy. It's not a simple copy and paste job. The road from a brilliant idea to a successful, healthy transgenic animal is absolutely full of hurdles and challenges.
For starters, the whole process is incredibly inefficient and super expensive. We're talking very, very low success rates. And sometimes you do all this work, the gene gets into the DNA perfectly, and then it just doesn't do anything. It doesn't turn on. Even when you do get it to work, it can take 10, maybe even 20 generations of careful breeding to create a stable line of animals that reliably has that trait. And on top of all that, you're messing with the fundamental code of life. There's always the risk of unexpected side effects.
Because think about it, for any of this to really be useful on a large scale, the change can't be a one-time thing. The holy grail, the ultimate goal, is what scientists call germline transmission. That just means making the new gene a permanent part of the animal's reproductive cells, its sperm or eggs. That's the only way to make sure the trait gets passed down reliably to all of its kids and their kids, and so on.
So, from glowing mice to life-saving medicines, it's clear that transgenesis has unlocked possibilities that really were just science fiction a few decades ago. But as our power to literally rewrite the code of life gets stronger and more precise, the questions we have to ask get bigger and more profound. The potential is absolutely immense, there's no doubt about it. But the real challenge for all of us is figuring out how we navigate the complex scientific and ethical landscape that comes with this incredible power.